Welcome to BioTechBuilder. I'm excited to show you one of my favorite labs, which helps reinforce the principles of pipetting, which will help students learn not only important skills for the laboratory, but will also prepare them well for some microcredential testing that they can do at the end of our course. So come on in, lab, and I'll show you what we got.
The procedure for this experiment is not particularly hard, and it should be reasonably familiar for students who have been in the lab for any amount of time, really. And it is to simply add a weigh boat to the balance, tear the balance, add a tip to the pipette, and then precisely measure 200 microliters in my case to weigh that volume, which should be 0.2 grams if the pipette is properly calibrated and the pipetting has been done accurately. This procedure, the students are asked to repeat it 6 to 10 times and then gather data for carrying out a calculation related to the accuracy of their pipette. All right, so I'd like to go through the calculations associated with this lab activity, partially because I think that's where the connection between the work that we do in lab and the consequences that that work has for our experiments and the skills that we bring to the lab bench is actually made more clear. So what I've done is listed here 10 trials of that pipetting activity with the sample masses that were measured. All the water was at 23 degrees Celsius. And there are two calculations associated with completion of this lab activity. The first is to calculate the average volume of water that was dispensed when we carried out all these measurements. So that calculation, the average volume of water that was dispensed, is a product of the average weight of the water that we measured and a factor called a Z factor, which is related to the water at a particular weight. It's a factor related to temperature of the water. So when I carry out the calculation to determine the average of these masses, that turns out to be 0.197. And when I look at the table in the lab protocol for the Z factor at 23 degrees, that turns out to be 1.0035. And so the average weight of the water that we've measured when you carry out this multiplication is 0.197. And so that brings us to the second calculation that we have to carry out, which is the percent accuracy of our pipettes. The percent accuracy is a calculation of this, that average that we just measured over the volume that was keyed into our pipettes. This volume has to be entered here in milliliters times 100% to get a percent average. So with this calculation, I take this number 0.197 divided by the volume that was put into our pipettes, 0.200. That's in milliliters times 100%. And it turns out that my pipette was 99% accurate as I carried out this experiment. I hope that's pretty clear, and I hope the connection between the work that we do in lab and the meaning that it has in terms of the skills that we bring to the lab and the quality of our equipment is more clear.
Well, I'm so glad you've watched all the way to the end. I hope you feel like this is something that you might like to do with your students and at your school. If it is, we are sending out a couple of sample kits and you can contact us to get one. Let me show you what's inside.
The protocol that can be downloaded from the website or that will come in the sample kit includes all the instructions related to accurately pipetting as well as carrying out the calculation for accuracy of the pipette. So there's some numeracy, there's some skills training associated with this lab that is hard to really convey when you simply look at the procedure of it. And then in this sample kit, there is a weigh boat, a beaker for water, along with some tips. The tips fit this fixed volume pipette. This is 100 microliter mini pipette. So you would expect the water that you measure to weigh 0.1 grams. There's a thermometer because the temperature of the water influences the mass that you'll measure, a couple of stickers, and a small balance that will help you carry out this experiment. I hope this seems enticing and something that you could teach, something that's valuable to teach. And we really do look forward to hearing from you.